This is an overview of an article entitled Tissue-Specific Differential Expression of Novel Genes in Long Intergenic Non-Coding RNAs in Humans with Extreme Response to Evoked Endotoxemia, published in the November 2018 issue of Circulation, Genomic and Precision Medicine. Cytokine responses to activation of innate immunity differ between individuals, yet the genomic and tissue-specific transcriptomic determinants of inflammatory responsiveness aren't well understood. There are likely to be many as-yet unknown genomic components that regulate tissue-specific responses and clinical outcomes in inflammatory disorders. Understanding tissue-level transcriptome responses to activation of innate immunity in humans has the potential to identify novel genes and pathways that modulate disease trajectories, which are not identifiable by studying static inflammatory biomarkers or through interrogation of non-human model organisms. Multiple tissues and cell types are involved in the response to pro-inflammatory stimuli and in the resolution of inflammation. In addition to key protein-coding genes, long intergenic non-coding RNAs, or lincRNAs, are emerging as important inflammatory transcriptomic modulators with diverse tissue-specific functionality and low evolutionary conservation relative to messenger RNAs, or mRNAs. The authors of the paper under discussion hypothesized that novel human tissue-specific inflammatory mRNA and lincRNA responses could be revealed through interrogation of evoked transcriptomes during experimentally controlled in vivo endotoxemia. In the Genetics of Evoked Response to Niacin and Endotoxemia study, healthy people underwent exposure to an endotoxin challenge, namely 1 nanogram per kilogram of lipopolysaccharide, or LPS. The authors selected the individuals with extremely high or low clinical inflammatory responses to LPS stimulation, defined as the top 5th percentile versus bottom 5th percentile of the febrile and plasma cytokine clinical inflammatory responses. These individuals were termed high responders or low responders. The authors then performed RNA sequencing, or RNA-seq, of CD14 monocytes and adipose tissue collected before and after administration of LPS. As indicated here, there are a large number of mRNAs and lincRNAs that were differentially expressed between high and low responders, with a larger number of differentially expressed RNAs in CD14 monocytes than in adipose tissue. There were substantial differences between monocytes and adipose in the high responders, speaking to tissue specificity. Further details are available in the online version of the paper. The authors assessed expression of 10 lincRNAs by qPCR in samples from 6 subjects who had undergone RNA-seq, that is, a discovery group, and in 6 additional subjects from the gene study who had high inflammatory responses, but were not in the RNA-seq study, that is, a validation group. As shown here, qPCR validated 8 out of the 10 lincRNAs as statistically significant in both groups, while the other 2 trended in the same direction. A similar analysis was performed with 5 lincRNAs in adipose tissues, with 4 of the lincRNAs being validated, as shown here. The authors then assessed for tissue-specific differentially expressed RNAs in high responders. As shown in the graph on the left, a lincRNA was confirmed to be downregulated in CD14 monocytes, but upregulated in adipose. This particular lincRNA is conserved in human and mouse. The graphs on the right show confirmation of 11 tissue-specific, differentially expressed mRNAs. The authors hypothesized that, like protein-coding genes, lincRNAs highly responsive to LPS stimulation are likely to be involved in mediating and regulating LPS-induced responses. They identified and validated one such lincRNA, which they named monocyte LPS-induced lincRNA regulator of IL-6, or MOLR-IL-6. This particular lincRNA is human-specific. the authors examined the expression of LPS-induced inflammatory responses in THP1 monocytes following knockdown of MOLR-IL-6 and observed a reduction in IL-6 expression, as shown here in Panel F. Further details are available in the online version of the paper. In conclusion, the authors observed clear differences in the magnitude of the transcriptional response post-LPS in CD14 monocytes and adipose between high and low responders, identified and validated a number of tissue-specific, differentially expressed lincRNAs post-LPS, and characterized MOLR-IL-6 as a novel, non-conserved, human-specific lincRNA that enhances LPS-induced IL-6 expression in monocytes.